All right. Hello class. Today my paper will be on, I'm at camera presentation actually. Let me just share my screen. So my presentation on the biorender is on mechanosensitive pathways involved in the cardiovascular development and homeostasis in zebrafish. This is the paper I chose. So just some quick facts on zebrafish. So actually phylogenetically, zebrafish and humans aren't very much alike, but some well, like for example humans and sharks, they have similar when they're actually infants. When humans are infants, they have similar cardiovascular systems to sharks. Well, it turns out that humans and zebrafish also have vast similarities in their genetics and cardiovascular systems. Now zebrafish are actually very common when it comes to studying genetics and diseases because well, like I said, they have similar cardiovascular systems and genetics. So what better way to test diseases and not diseases, but like test ways to regenerate tissue, for example, or regenerate heart, which is a tissue. But stuff like diseases, you know how to combat them better when to than using a model that's very similar genetically and systematically to humans. So some important aspects about zebrafish that actually make them so special and such important models is that zebrafish have they have the ability to repair vascularly, and they have the ability to regenerate their heart. So this is very important for scientists because obviously we want to make breakthroughs in science and medicine. So what better way to make a breakthrough than be able to say that, oh, okay, we found a way for you to regenerate tissue in your heart that would save millions and millions of people, because we know that heart diseases and heart problems are probably like killing every other person probably dies from a heart disease. So if we can find a way to eradicate that and eliminate that and find a way to regenerate hearts or tissue or regenerate arteries or scarred arteries, stuff like that, that'd be very important. So they actually noticed that in the zebrafish models that shear stress actually activates mechanosensitive pathways. And the proteins that are actually activated are Notch1, protein kinase C, isoform, and other proteins. So shear stress and hemodynamic shear stress. So here shear stress is the force caused by blood flow. So if you have a very intense blood flow or very ramped up blood flow, it can cause well, it depends on what you have, right? So usually you have veins. Veins are very narrow, so not a lot of blood flow. Veins can't really deal with a lot of force generated by blood flow. But you have arteries. Arteries are thicker, right? So arteries have they have better they have mechanical qualities, right? So they can be fluid-like or solid-like. So that's what shear stress is force caused by blood flow. Now, NOSH1, which is one of the proteins that I mentioned earlier that is activated by shear stress, is stress sensitive. So that makes sense, right? It's activated by shear stress because it's stress sensitive. Now, hemodynamic shear stress now this is a frictional force from blood flow, but more specifically, it's acting on the vascular endothelial cells, which also play a huge role in the development of vascularly and homeostasis. Now, mechanosensitive pathways. These pathways are what basically control and where vascular development is actually happens and where regeneration happens. So without mechanosensitive pathways, we wouldn't be able to. Well, a zebrafish wouldn't be able to regenerate and develop that regeneration vascularly and regenerative in tissue wise. So this is the diagram and drawing that I try to recreate my biorender. So you can see how I didn't go this specifically into detail, but you can see how like there's I think that's notch 1, and down here it explains in the figure what exactly is going on. But if you look on the bottom right, there's regeneration, top right amputation, bottom left impaired SIV, and top left is SIV. So you have glycosis, you have the nucleus, the ethyl cell we saw earlier, which is important in regenerating vascularly. So there we go, notch one, which is shear stress. So I'm assuming this red line right here is blood flow, shear stress, blood flow right here. So which is actually activating these proteins right here. All these proteins right here are being activated by shear stress, which is causing regeneration and in tissue, and which is causing vascular development and regeneration also as well. So now let me show you my attempt at remaking this. So in the biorender, it didn't let me like make that elaborate of a diagram, but this is a zebrafish heart. This is stress. This is an artery that has gone shear stress. This is a red blood cell, and I have a picture of two guys, one that's bald and one that has hair. So just kind of showing that regeneration aspect. So like we said earlier, zebrafish are very similarly biologically and cardiovascularly to humans. So scientists like to use them for models for regenerative purposes and diseases models, experiments, stuff like that. So zebrafish, like I said earlier, have found to be they can regenerate their heart tissue and regenerate vascularly. So we've been looking into that as to find a way that maybe we can apply this to humans and try to save some hearts ourselves and some tissues ourselves. So like I said earlier, we have the zebrafish heart, which has that qualities of regenerating. You have stress, and so when shear stress, well, in this case for zebrafish, it would be hemodynamic stress. Shear stress actually activates and provokes the mechanosensitive pathways to be activated, which then causes the proteins that are shear stress sensitive to be activated, and that's what causes the regeneration to actually happen. So that's basically all I have for today. Thank you for attending my presentation.